Here we're looking at the light microscope. This is a hugely important piece of equipment in medicine, often used to diagnose certain pathologies, looking at tissue samples to identify a specific cancer or tumor. So today we're going to look at the parts of the microscope and how to use it. Toward the latter end of this video, I'll show you the very fundamental way to use the microscope, making it the easiest for you to obtain the tissue sample in your field of view, and then ultimately zoom in on that tissue sample to find exactly what you're looking for. So when we look at this microscope, here you can see the base. The base will actually house the light unit, which is right here. So I'll turn that on. So there's your light source illuminating up through your tissue sample. This is your light intensity control. So you want to be careful when you're using a light microscope, because realize it's projecting light from underneath up through a tissue sample. That tissue sample is usually just one cell thick or so, and that allows the transfer of light, which will then allow you to see the stained tissue sample on the slide. So as things get magnified, so does the light. That intensity will get amplified. You don't want to burn your retina. So always start out with the light at a very low intensity. I'm going to turn it off here now. So if you look, this is the arm of the scope. And the arm of the scope is basically going to connect the base of the scope to the ocular lens and the objective lenses. This ocular lens universally is 10 power. That's pretty universal. Now, this can be a single ocular lens or binocular, depending on the style of microscope you're working with. Right here, this is called the nose piece, or sometimes referred to as the turret. And it houses the objective lenses that you can see here. Right now, I have the scope set to its lowest power, and that's how it should be when you're starting out. You always want to be on the lowest power so that you don't crash your slide into your lens. Right here is a stage clip, and that's what we're going to put our tissue sample into. So you take your slide, pull this to the side. That allows you to place the tissue sample in there, the slide, and then release the clip, and it holds it in place. When you're actually looking for your specimen, again, start out on your lowest power, because that's going to give you your largest field of view, which means it's going to be the easiest for you to locate where that tissue sample is on the slide. Right here, there are adjustments for this entire stage. It'll move that stage forward and backward so you can advance or retract the specimen in your field of view. And this one here, the lower one, moves the clip assembly from side to side. Now, when you're viewing in this microscope, realize that when you adjust the stage or the clip assembly, front to back or side to side, it will appear to move in the opposite direction in your field of view. So you always want to start out on your lowest power. Once you bring that into focus, and right here is your course adjustment. So this will bring the stage up rapidly, and when you're on the lowest power, that's just fine. You can bring it right up. Once you get that low power into focus, after that, you'll probably just be able to use the smaller knob, which is your fine focus, for each of the higher power lenses. So as you turn them to that power, you'll just fine focus your specimen, and then this would be the highest power. Same idea. You'll just bring it into focus using the fine adjustment. If we look underneath the stage, here we have what's called an Abbey condenser. An Abbey condenser. And it has this lever here, which operates as an iris diaphragm. Okay, what that does is it creates contrast. So you have your illumination coming up, and as you close this down, which would be to move it from left to right, it creates contrast. That contrast will allow you to see greater detail in your tissue specimen. So now let's take a look at this tissue specimen, and I'll show you what I was mentioning earlier about starting with the lowest power and making it easier to find your tissue sample on the slide. Okay, so here's our tissue sample. And this is actually taken from a cat trachea. So their trachea is constructed much like ours. It has hyaline cartilage, smooth muscle, areolar connective tissue, and pseudostratified columnar epithelium. That last mentioned, pseudostratified columnar epithelium, that's the tissue we want to see. If you look at this, we're in our largest field of view because we're set at the lowest power. And there's two reasons why you want to always start at the lowest power. One, to ensure that you don't crash the slide into your lens and damage both, which could be very expensive. And then two, it gives you the largest field of view. It makes it the easiest to acquire your tissue sample. So that's what we're seeing here. Our tissue sample at the lowest power provided the largest field of view. But what the pointer is actually on right now is the smooth muscle. So I'm telling you this because I want you to recognize that when you put a tissue sample in and you look at it at the lowest power, I don't want you to assume that the first thing you see is the tissue you're looking for or what you're expected to see. You're at low power. Now, low power might be optimal depending on what tissue type you're looking at. But for this tissue type, it's not optimal. It's just to help us acquire our sample. So let's dial up now to the next highest power. Before I do that, again, the pointer is on smooth muscle. Beyond that is the hyaline cartilage, and then the connective tissue and the columnar tissue. So let's move up. Now you can see that we're on the hyaline cartilage, the band of hyaline cartilage. And the smooth muscle is that pink band just below. I'll back this up. So there's your smooth muscle. Here's your hyaline cartilage. This is connective tissue, and to the right of the pointer, some glandular tissue. And right there, that is the tissue sample we want to look at up close and in detail. That's our pseudostratified columnar epithelium, but it's still at a lower magnification than we need to really observe this properly. So we'll dial this up now to the next highest power. And of course, as you go up in power, it kind of changes your field of view relative to your pointer, and that's simply a result of the magnification effect. So there you can see we're beyond our tissue sample. But we'll back up here, and there's our pseudostratified columnar epithelium. Back here is the hyaline cartilage and the smooth muscle before that. So let me see if I can bring this into focus a little better. Maybe create a little contrast here. Okay, so it's not the best, but you can see the cilia on top of the cells. So that's what we're looking for, this tissue sample at the end of the pointer. But again, there are 4 different tissue types in this one sample of tissue on this slide. So you want to start again at the lowest power to give yourself the best chance of success in locating what you're trying to find and focusing it in properly. When you start at the lowest power, you can focus with the coarse adjustment. Bring it right up and bring it into focus. At that low power, you will not crash the lens. After you've focused at low power, each power after that, you'll probably only require the fine adjustment to bring it into focus. So there you go. There's just a simple example or demonstration of how to use the scope properly to ensure your best chances of success. Finally, at the end of lab, you always want to turn your nose piece to the lowest position, lowest power lens, or even this 4th position, which is often vacant because it's used for oil immersion. But if not, then use the lowest power lens, turn it to that position, remove your specimen slide from the stage, and then ultimately turn the stage to its lowest position, dialing it down with the coarse adjustment, just like that. Finally, take the cord and you want to wrap it around the base of the scope for storage, and then place the scope back in its proper placement in the microscope cabinet. And there you go. So that is the light microscope, the parts and fundamental use of this scope, the easiest way to locate your tissue sample within the slide.